Hi everyone. So this week you will have already had started with the PowerPoint on the immune system and the lymphatic system, and then I'm just going to start in on the first maybe half, maybe slightly less than half of this heart lecture, and then we'll pick up the rest of it next week. The heart is approximately the size of a closed fist, and it's located in a region in the chest or thorax known as the mediastinum, which is between the second rib and the fifth intercostal space, basically like between the fifth and the sixth rib. Excuse me. It's also located just left of that mid-sternal line. So you see more of it to the left of the sternum than the right. And it's enclosed in this double-walled fibrous sac known as the pericardium. The pericardium has two layers. There's the superficial fibrous pericardium, which has the consistency of almost like onion skin. It's very tough, and this prevents and protects, anchors, and prevents the heart from overfilling. It's a nice like protective boundary to keep it within the confines. The pericardium has an inner serous, right, much thinner, sort of lubricated, two-layered serous pericardium. So there's two layers to this. The parietal layer, think parietal always lines like a cavity or a wall of something. So you have that outer fibrous, very thick onion skin-like pericardium, and then lining the inside of that is the slippery, thin parietal layer of the serous pericardium. The visceral layer, also known as the epicardium, is the serous layer that's right on the external surface of the heart itself. And the visceral and parietal serous pericardium are separated, by a fluid-filled pericardial cavity. Think of it as it decreases friction, right? As the heart contracts and expands, it prevents those layers from rubbing upon one another and giving us pain. When this happens, or rather when there is a swelling and an irritation of the pericardium, it can cause extreme chest pain as well as sometimes other symptoms. And the causes of pericarditis, which I should have prefaced it, it's called pericarditis. The causes of this are varied. So it includes viral and bacterial infections as well as post-infarct pericarditis, which is post-heart attack. If we look at the layers of the heart wall, we have the outermost layer known as the epicardium. This is a visceral layer of the serous pericardium, right? One and the same. That visceral layer is also known as the epicardium, and it's the most superficial or external layer of the heart. The myocardium makes up the bulk of the heart. It's the muscular layer, and it's comprised of bundles of cardiac muscle cells, and it also contains the fibrous skeleton of the heart. So it's that interlacing, irregular connective tissue that gives the heart the ability to stay together despite having to beat constantly throughout our life. And then most internal is the endocardium, which is the layer of the heart that comes in contact with the blood and actually continues on to become the endothelial lining of the blood vessels. And it's important to note, while this is a cartoon, that the way that the cardiac muscle bundles are arranged is much different than we see in skeletal muscles, right? It's kind of like if you wring out a towel. You do it in that circumferential motion where you're twisting in the opposite directions. And when you think about the heart, it has to do the same thing, right? The heart doesn't contract up and down to become shorter and longer. Instead, it wants to squeeze as much blood as it can, as much as it's supposed to, out to get it out to either the lungs or to the rest of the body. The heart is comprised of 4 chambers. The 2 superior chambers are known as the atria, and they're separated internally by something called the interatrial septum. And then we see external manifestations or expansion of the heart, of the atria, known as the auricles. They look like little ears. And these are sort of the overflow. If needed, they can increase the blood volume of the atria. And then the 2 inferior chambers, and much larger and more muscular, are known as the ventricles. So the atria are sort of the collecting chambers, where the ventricles are the pumps. The ventricles are separated by the interventricular septum, and then the anterior and posterior on the external side of the heart there's an interventricular sulcus, and this kind of marks the delineation between the left and the right ventricles on the outside of the heart. In terms of the atria, as I mentioned, they're the receiving or collecting chambers. Their walls are ridged by something called pectinate muscles. The vessels that enter the right atrium are the superior vena cava, and dumping venous blood from the upper part of the body, the inferior vena cava from the lower parts, and then the coronary sinus, taking the blood from the heart itself, the deoxygenated blood. The vessels that enter the left atrium are the right and left pulmonary veins. And I want to I wanted to just point out that for the most part, veins are carrying deoxygenated blood and are moving towards the heart, and then arteries are carrying oxygenated blood away. So with the pulmonary veins, they're adhering to the rule of returning to the heart, but they're the only ones that are carrying oxygenated blood. The ventricles are the discharging or pumping chambers. Their walls are ridged by trabeculae carnae, and there are papillary muscles that project into the ventricular cavities. The vessel that's leaving the right ventricle is the pulmonary trunk, and I know I botched this on the question for the homework. The vessel that's leaving the left ventricle is the aorta. I think I just stated aorta twice. The heart is basically two side-by-side pumps. The right side is the pump for what's known as the pulmonary circuit, meaning that the vessels that carry blood to and from the lungs. The veins, like I said, go towards the heart, with the exception of the pulmonary veins. They do also return to the heart, but they're carrying they're carrying oxygenated blood. The left side is the pump for what's known as the systemic circuit, and these are the vessels that carry the blood to and from all the rest of the body tissues. And again, artery away from the heart. So if we look at the pathway of blood through the heart, if we start at the right atrium, right, we have the deoxygenated blood returning from the superior vena cava, the coronary sinus, and the inferior vena cava. It passes through the tricuspid valve and goes into the right ventricle. Now the blood in the right ventricle, keep in mind, is lower in oxygen. So where do we want to send it to get reoxygenated? We want to send it to the lungs. So from the right ventricle, we have the blood go through the pulmonary semilunar valve into the structure known as the pulmonary trunk, right here, which then splits into the left and right pulmonary arteries. Some folks, I worked in pulmonary studying this specifically, so the debate is alive and well, whether we call it the main pulmonary artery and then the left and right pulmonary arteries, or we call it the pulmonary trunk. I think it's easier to call it the trunk, but whatever. And then these pulmonary arteries travel into smaller and smaller vessels into the pulmonary capillaries and pick up oxygen in the lungs. Excuse me. Now, from the lungs, after the blood has picked up oxygen, it returns to the heart through the pulmonary veins. These are the one exception, right? They're veins, they're returning to the heart, which is in line, but they're carrying oxygenated blood. This oxygenated blood enters the left atrium. From the left atrium, that oxygenated blood passes through the bicuspid valve into the left ventricle. From the left ventricle through the aortic semilunar valve, into the aorta, and then from the aorta into the systemic circulation to the rest of the body. The coronary circulation, oftentimes we forget about this. The heart is constantly working. It never gets a break or we'd be dead. So it needs constant blood supply and blood renewal. So the functional blood supply to the heart itself is known as the coronary circulation. Arterial supply contains many anastomoses or junctions. This is sort of a fail-safe, right? You'll hear about people that have like a certain percentage blockage in their heart, and while they may have had a myocardial infarction, in most cases it's discovered before they do. They might be having like chest pain. The reason for all these collateral routes is that the heart has to keep going, right? So at all costs, we hope that even say this right coronary artery is blocked, it can go through one of these, like the right marginal artery, to get across, to get blood flow to these regions. The arteries of the coronary circulation are the right and left coronary arteries. which run in that atrioventricular groove on the external surface of the heart, as well as the anterior interventricular arteries. The veins are the small cardiac vein, the anterior cardiac vein, and the great cardiac vein, which is on the posterior side of the heart. You can see it sort of transparent here. This is just a nicer, more sort of blown up view. This is the posterior surface, by the way. You can see the coronary sinus very well on the back between the atria and the ventricles. And there's also a link in the static PowerPoint to watch a really good video on this. In terms of homeostatic imbalances of the heart, angina pectoris is thoracic pain caused by a fleeting deficiency of blood delivery and therefore oxygen delivery to the myocardium. People kind of call it agita sometimes. And then a myocardial infarction, my apologies, I can't go back at this point, is when a part of the heart becomes blocked to its blood supply altogether, and because the heart is devoid of oxygen, that part of the heart, those myocardial cells die. And remember, muscle tissue and nerve tissue doesn't really regenerate, so it causes permanent damage to that region of the heart. Heart valves help to ensure unidirectional blood flow through the heart so that we're not having backflow and having deoxygenated blood mix with oxygenated, or backflow so that the heart kind of blows up and we don't get efficient blood supply to the body. The atrioventricular valves prevent backflow from the ventricles back into the atria, so they're between the atria and the ventricles. On the right side of the heart, we have the tricuspid valve that has 3 flaps. On the left-hand side, we have the mitral or bicuspid that has 2 flaps. On the bottom part, the posterior surface of those AV valves, we have something called chordae tendineae, aka heartstrings, that anchor the cusps of those AV valves to the papillary muscles which are embedded in the ventricular wall. The semilunar valves are between the ventricles and either the pulmonary trunk on the right or the aorta on the left. So we have the aortic semilunar valve and the pulmonary semilunar valve. If this is a top-down view of the heart, so we can see the vessels have been cut. So we see the tricuspid valve here on the right, the bicuspid or mitral on the left, and then we see those semilunar valves. You can see the valve, the cusps kind of look like half moons. This is a great picture showing, right? This is the inferior surface of one of the AV valves. Here are those chordae tendineae or heartstrings, and they're attached to the papillary muscles which are embedded in the ventricle wall. So when the ventricle contracts, it pulls down on these chordae tendineae, which pull on those flaps and close them so that there's not backflow into the atria above. This is a sagittal section. Actually, I take that back. This is a frontal section of the heart. So you see the interventricular septum. And important to note is the left ventricle has to pump blood under high pressure to all of the body, down to the tip of your big toe, whereas the pulmonary circuit is only sending blood to the lungs, which are right next door to the heart. And we want the blood to go under low pressure and go a little slower through the lungs. 1, so it doesn't rip apart the pulmonary capillaries, but 2, so it allows for adequate time for the blood to pick up oxygen. So because of that, you'll look at the free wall, right, the outer wall of the right ventricle as composed to the left. The left ventricular free wall, the left ventricular outer wall, will always be much, much thicker and more muscular than that on the right in a healthy heart. Walking through the flow of blood through the heart by starting with blood returning to the heart into the atria, those uppermost receiving chambers. At this point in time, if you look at the diagram, you see that the AV valves are closed. So as blood is passively filling in the atria, right, pressure is building against these or behind these AV valves. Eventually, the pressure builds enough that these AV valves are forced open, and blood initially passively starts to fill the lower chambers known as the ventricles. Eventually, those AV valves will snap shut, and at this point in time, the semilunar valves going into the pulmonary trunk and the aorta, respectively, also remain closed. So when we go on in this lecture, we'll talk about isovolumetric contraction and isovolumetric relaxation. Anytime you hear the term isovolumetric, iso means same or equal. So for that to happen, all valves have to be closed. So at this point here, we get the last little bit of contraction of the atria, pushing the blood into the ventricles, and then all 4 valves close. Okay? The ventricles start to build up pressure, and when they contract, eventually, enough pressure builds that it forces open the semilunar valves. And the semilunar valves, let me go on to the next slide, they will open, okay, forcing blood into the pulmonary trunk or the aorta. And then we kind of start to resolve things. So the ventricles relax, intraventricular pressure falls. We don't want backflow, so the AV valves are still closed, and then those semilunar valves close. And keep in mind that things are happening concurrently, meaning that while all this is happening, the blood is passively returning to the atria via the superior vena cava, the inferior vena cava, and the coronary sinus. Compare the microscopic cellular anatomy of cardiac muscle to skeletal muscle. They're both striated, but unlike skeletal muscle where the cells are long and cylindrical and run very sort of flush and parallel to one another, cardiac muscle cells are branched. They also have a different T-tubule and sarcoplasmic reticulum structure. So if you remember, the T-tubules are the invaginations or the dippings in of the muscle cell membrane so that the electrical current, the nervous impulse, can sweep down and open those voltage-gated channels on the sarcoplasmic reticulum. In skeletal muscle, we want the muscles to contract, the different motor units to contract asynchronously to prevent muscle fatigue, whereas in cardiac muscle, we want all of the cardiac muscle cells to contract synchronously, to have that wholehearted contraction. Similar to skeletal muscle, because muscle in general is very metabolically active, cardiac muscle has a large number of mitochondria as well. Important feature of cardiac muscle that you may remember from A&P1 is the presence of intercalated discs between adjacent cells. Intercalated discs are these densely, darkly staining areas between connected cardiac muscle cells that are they're areas between cells that are very highly enriched with desmosomes, which are the cell-cell junctions or intercellular junctions that basically function like buttons and snaps on a jacket, as well as gap junctions, which if you think of like a paper towel tube or a toilet paper tube sticking out, through one cell membrane, and then the same thing sticking out of the adjacent cell membrane, so they interconnect and basically form a tunnel where you can have direct passage of cytosol and small dissolved substances directly between cardiac muscle cells. So these intercalated discs are regions where there are a lot of gap junctions and a lot of desmosomes, because remember, the heart is contracting continuously throughout our lives, so we don't want those cardiac muscle cells to physically rip apart. The whole function of having these intercalated discs, especially with the gap junctions, which allow direct communication, direct cytoplasmic communication between adjacent cells, is all aimed at having the heart behave as what we call a functional syncytium, meaning that all of the cells of the atria contract simultaneously, and then with barely any time to spare, then go all the cells of the ventricles. Whereas, like I mentioned in previous slides, in skeletal muscle, unless we're running for our lives and we want to max out on that muscle, instead we will have some fibers of the skeletal muscle excited and contract, and then others to prevent muscle fatigue. The heart does not have that luxury. We also will examine the depolarization of cardiac muscle as opposed to what you covered with skeletal muscle in A&P1. So depolarization or activation of the heart is rhythmic and spontaneous. How is it spontaneous? Well, about 1% of cardiac muscle cells are what we call autorhythmic, meaning that they are self-excitable. They are able to stimulate an action potential without any nervous innervation. Gap junctions then ensure that the heart contracts as a unit, as a functional syncytium, by allowing that cell-to-cell communication rather than having a cellular molecule go across the cell, bind to a receptor on another cell, and have to propagate a response. With gap junctions, there's literally direct communication. Cardiac muscle cells also have a very long absolute refractory period. So if you recall, the refractory period, the absolute refractory period, is the period during which a muscle cell cannot be stimulated to contract again, regardless of the strength of the stimulus. Why is this particularly important in cardiac muscle cells? If you compare them to skeletal muscle, right? Yes, you might get a Charlie horse, or you might get such a contraction of a skeletal muscle if it's stimulated before the end of its refractory period. That may be worst case if somebody falls down or, God forbid, crashes their car because they're driving, right? But however, if the heart is stimulated to contract before it's able to relax, somebody goes into fibrillation, right? So this is important because somebody can die. Their heart can literally stop functioning correctly if those cells get stimulated again too soon. So depolarization of cardiac muscle opens voltage-gated, what we call fast sodium channels in the sarcolemma. The sarcolemma, again, is just the name for the muscle cell membrane. This entrance of positively charged sodium causes a reversal of membrane potential. So the voltage inside the cell goes from a resting membrane potential of minus 90 millivolts to positive 30. We see a depolarization wave sweep down from the surface of the sarcolemma into those T tubules, which then causes the sarcoplasmic reticulum to release calcium. The depolarizing wave also opens what we call slow calcium channels in the muscle cell membrane. So a combination of release of calcium from the sarcoplasmic reticulum storage, as well as entrance of calcium from outside the muscle cell, causes a calcium surge. And this causes a longer depolarization phase than you would see in a skeletal muscle. Okay, you see that plateau down here with the number 2. This calcium influx triggers the opening of additional calcium-sensitive or calcium-responsive channels in the sarcoplasmic reticulum, which liberates greater bursts of calcium. So the excitation, right, the stimulation by an electrical current, coupled to the contraction, occurs as calcium binds to troponin, similar to what we see in skeletal muscle, and it pulls tropomyosin off actin, and the actin and the myosin can begin to interact. The duration of the action potential and the contractile phase is much greater in cardiac muscle than it is in skeletal muscle for the reasons I stated on the previous slide. Repolarization results from inactivation of calcium channels and the opening of voltage-gated potassium channels. We mentioned that the heart, there are about 1% of cardiac muscle cells that are autorhythmic. They can spontaneously contract and stimulate an action potential. Or excuse me, they can spontaneously depolarize. And this plays into the greater intrinsic cardiac conduction system. So the intrinsic, right, means within the heart itself. Cardiac conduction system is a network of non-contractile but autorhythmic, spontaneously depolarizing cells that initiate and distribute impulses to coordinate the depolarization and contraction of the heart. These autorhythmic cells have an unstable resting membrane potential. It's called a pacemaker potential, due to having these unique slow sodium channels that just stay open. At threshold, calcium channels open, meaning at threshold, enough sodium comes in regularly and rhythmically through these chronically open slow sodium channels that it causes calcium channels to open, and we get that explosive calcium influx that we talked about in the previous slides. And again, repolarization, resetting, results from inactivation of those calcium channels and the opening of voltage-gated potassium channels. So now let's walk through the various components of the intrinsic contraction system of the heart. It starts with the sinoatrial or SA node in the wall of the right atrium. And we call the SA node the natural pacemaker because it generates impulses at about 75 beats per minute or times per minute, which is one's average resting sinus rhythm, right? It's someone's average healthy pulse, 70 to 75 times a minute or 70 to 80 beats a minute. Think about if you've ever run, like for sports drills, and you run in a single file line with the person in the front leading the pace, right, and the person in the back has to run and catch up. You know that the person in the front is leading it, they're setting the pace. So the sinoatrial node depolarizes faster than any other part of this myocardial conduction system. Therefore, it sets the pace. It is the pacemaker. So we see it up here with number 1. Okay. The atrioventricular, or AV node, has smaller diameter fibers and fewer gap junctions, and it delays the impulse conduction from the SA node to the ventricles because of these factors. It delays it by about 0.1 seconds, a tenth of a second. It also depolarizes at roughly 50 times per minute as opposed to the 75 of the SA node. So if the SA node were damaged in any way and couldn't function, then the next pacemaker of the heart becomes the AV node, but then the heart rate, the pulse, drops from 75 to 50 beats per minute. Next come the atrioventricular or AV bundle, or otherwise known as the bundle of His. This is the only electrical connection between the atria and the ventricles. So if something were to happen to the AV bundle, it leads to a condition known as complete heart block. Then come the right and the left bundle branches, which you can see here going down each side of that muscular intraventricular septum. So they're carrying impulses towards the apex of the heart, this bottom pointed part. And finally, we end with the Purkinje fibers that sweep upwards along the outer walls of the two ventricles. The AV bundle, or bundle of His, and the Purkinje fibers depolarize at only 30 times per minute. So if we were to lose AV node input, right, say the SA node is already damaged, then the AV node gets damaged, the heart would only beat 30 times per minute, which, as you can imagine, would not be sufficient to keep functioning as normal, so there would be physiologic consequences. So if we look here, just to summarize, we have the SA node up here in the right atrium, generating about 75 beats per minute. Impulses then get transmitted to the AV node, where there's, because of fewer gap junctions and smaller diameter fibers, there's about a 0.1 second delay in impulse conduction. From the AV node, we have the electrical impulse come down the AV bundle, or bundle of His, down each of the bundle branches going to the right and to the left ventricles, and then the Purkinje fibers bringing that impulse from the apex of the heart up through the outer walls or free walls of each ventricle. Now what happens, as you know, even maybe without having studied this before, we hear with loved ones, with friends, that something happens to their heart rhythm and it can be catastrophic in certain instances. So defects in the intrinsic conduction system of the heart may result in arrhythmias or irregular heart rhythms, uncoordinated atrial and ventricular contractions, and fibrillation, which is rapid irregular contractions of the heart, which becomes useless for pumping blood. A defective SA node may result in what's called an ectopic focus, meaning that an abnormal pacemaker takes over, whether that be the AV node or the AV bundle, etc. If the AV node takes over, there will be a slower rhythm, 40 to 60 beats per minute, which is not good for maintaining bodily function. However, worse than that, as I mentioned, is if something happens to the AV node, because then the transmission of the impulse can't go from the atria to the ventricle, so this results in either a partial or a total heart block. And this is when few or no impulses from the SA node reach the ventricles. There's also extrinsic innervation of the heart, right? The heart has its own intrinsic system that self-regulates it. However, we know when you get nervous, when you get excited, when you're sad, right, the heart rate will change. So there's outside innervation. The heartbeat is modified by the autonomic nervous system. There are cardiac control centers located in the medulla oblongata of the brainstem. There's the cardioacceleratory center that innervates the SA node and the AV nodes. It also innervates the heart muscle itself and the coronary arteries, all through sympathetic neurons, right? The ones that kick in through the fight or flight response. Oppositely, there is a cardioinhibitory center that inhibits the sinoatrial or atrioventricular nodes through parasympathetic fibers within the vagus nerves. And remember, the parasympathetic is the rest and digest part. An electrocardiogram, with the abbreviation either ECG for the American spelling or the EKG for the international spelling, is a composite of all the action potentials generated by both the nodal cells, right, the SA node down to the Purkinje fibers, as well as the contractile cells at a given time. And this results in three waves. A P wave, which corresponds to depolarization or activation of the SA node, the QRS complex, which corresponds to ventricular depolarization, and the T wave, which represents ventricular repolarization. So here we see that when the atrium is stimulated to contract, we have atrial depolarization creating a P wave. We also can measure the distance from the beginning of that atrial activation to its end when ventricular activation starts. And then we have the QRS complex. And if you look at the height where the R is, look how much greater in magnitude the activation or depolarization is from the ventricles versus the atria. Why? Because the ventricles are the larger, more muscular pumping chambers of the heart. And then we have the T wave, which represents ventricular repolarization or relaxation. If you notice, we have a wave for atrial dipole, ventricular dipole, and ventricular repole, but you don't see one for atrial repolarization. That's because that's happening during the QRS complex, and the QRS complex is so large in magnitude that it masks the atrial repolarization wave. Okay, so I will not bore you to death going through all of this, but I would recommend that you walk through, and these are color-coded to show you which parts of the heart are depolarizing and which are repolarizing at a certain time. Okay, and it corresponds to the wave form. And here is a EKG reading showing normal sinus rhythm, something called junctional rhythm, right? So this representation, if you look, this is definitely not a normal EKG, and the QRS complex, the T wave is off. So if you look, the SA node is non-functional. P waves are absent, and the heart is paced instead by the AV node. So the first waveform you see is the QRS complex. There's second-degree heart block, where some P waves are not conducted through the AV node, and hence more P than QRS complexes are seen. Right here you see a P wave, P wave, and then there's another P wave before you see a QRS complex. So you're literally missing ventricular contraction, which could be deadly. And then ventricular fibrillation, which is also could be deadly, where the ventricles are just depolarizing and contracting erratically and allowing no time to fill with blood, so the heart is not sending any blood to the body. And this has to be reset through electrical shock or defibrillation. When we think of heart sounds, the traditional lub-dub is what we associate it with the lub, or the first sound, resulting from the AV valves snapping shut at the beginning of systole or ventricular contraction, and then the second sound, the dub, being when the semilunar valves close at the beginning of ventricular diastole or ventricular relaxation. Heart murmurs are any abnormal heart sounds, most often indicative of a problem with either the AV valve or semilunar valves. A cardiac cycle refers to all the events associated with blood flow through the heart during one complete heartbeat. So this includes systole or contraction and diastole or relaxation. And I want to point out that it's sort of a convention, but it kind of isn't talked about, so one would not know that when we refer to systole or diastole, we have two sets of chambers, right? They're always referring to the ventricles because the ventricles are the pumps. So we start with ventricular filling, and this takes place in diastole. Why? Because the ventricles can only fill if they're relaxed and open. At this point, the AV valves are open, so blood, about 80% of the blood just passively flows by gravity from the atria into the ventricles. Atrial systole, or atrial contraction, occurs at the very end of ventricular filling, just delivering that remaining 20% or so of blood into the ventricles. The end diastolic volume, or EDV, is the volume of blood in each ventricle at the end of ventricular diastole, aka ventricular filling. Ventricular systole is when the atria relax and the ventricles begin to contract. There's rising ventricular pressure resulting in the closing of the AV valves behind them, and isovolumetric contraction takes place. So remember, in isovolumetric anything, all valves are closed. In the ejection phase, the ventricular pressure eventually builds so much that it exceeds the pressure in the pulmonary trunk and in the aorta, so it forces the semilunar valves open, and the blood goes into the pulmonary trunk or the aorta, and what's left is referred to as the end systolic volume, or ESV, the volume of blood remaining in each ventricle after ventricular contraction. And we obviously want there to be a large EDV, a large amount to fill, but a very small amount of end systolic. If we have a lot of blood left in the heart after forceful contraction, it means that something's not right, either too much pressure ahead or weak ventricular muscle. And then we have isovolumetric relaxation, and this occurs in early diastole. So again, all four valves are closed, the ventricles are relaxing, and meanwhile the atria above are filling with blood. Okay. So here we have the phases of an electrocardiogram, and you can see what's happening here. The P wave, you see it in dark pink here, and it's highlighted in dark pink up here. The atria are contracting. Then the QRS complex, the ventricles are contracting. And then the T wave, the ventricles are relaxing. Okay, so from the start of one P wave to the end of the T wave, that's one cardiac cycle. So again, phase 1, atrial contraction. This is initiated by the P wave. The atria contract, atrial pressure rises, and blood is forced through the open AV valves into the ventricles. Again, by this point in time, roughly 80% of the blood from the atria have passively just gone into the ventricles. This atrial contraction just forces the remaining 20 or so percent into the ventricles. The semilunar valves are closed this whole time, right? You see them boxed out in black, because if those were open, then the blood would just go right out. We want it to fill and to maximize filling in the ventricles and stretch those ventricular walls. Isovolumetric contraction then occurs. So the semilunar valves are already closed, the AV valves snap shut, and this leads to a rapid increase in ventricular pressure. Okay? Ventricular pressure continues to rise without a change in ventricular volume, hence the isovolumetric part. Phase 3 is rapid ejection. This is an initial rapid ejection of blood into the aorta from the left side, or into the pulmonary trunk or pulmonary artery, PA in this diagram, from the right ventricle. This whole thing begins when the intraventricular pressure that's been building during during isovolumetric contraction rises so much that it exceeds the pressures in front of those semilunar valves, and the semilunar valves open. The AV valves are closed at this time, so the atria are just passively filling, and that prevents backflow into the atria when those ventricles contract. Phase 4, reduced ejection, occurs with the start of the T wave. The ventricle muscles are starting to repolarize. There's a decline as a result in that ventricular squeezing, and ejection slows. The AV valves are still closed, but the semilunar valves remain open. Phase 5, now the semilunar valves close, the AV valves are still closed, so you get isovolumetric relaxation. Ventricular pressures continue to fall while ventricular volumes stay the same. They're getting ready to fill again. Okay, and then phase 6, now the atria that have been filling, because they've been filling and those AV valves were initially closed, it's going to force the AV valves open, and the ventricles start to fill rapidly with that 80% or so of blood that's just passively going in from the atria. And then phase 7, we've come full circle. The ventricles continue to fill with blood. Intraventricular pressures begin to rise. In normal hearts, so to speak, healthy patients, the ventricles are about 90% filled at this phase. The AV valves open. The atria are getting ready to contract again and start another cycle. Okay. Now, cardiac output, we're going to go through some important clinical parameters to note. Cardiac output refers to the volume of blood pumped by each ventricle in 1 minute. Cardiac output is equal to heart rate, or number of beats per minute, times the stroke volume, or volume of blood pumped with each beat. At rest, cardiac output should be the result of roughly 75 beats per minute in terms of heart rate, with an average stroke volume of about 70 mils per beat, resulting in a cardiac output of the heart of about 5.25 liters of blood per minute, which is an astounding amount when you think about it. It's 2.5 soda bottles. Maximal cardiac output is about 4 to 5 times one's resting cardiac output in non-athletic people, and can actually reach as high as 35 liters per minute in well-trained athletes. The cardiac reserve is the difference between one's resting and maximal cardiac output. So the cardiac reserve will obviously be greater in conditioned individuals. Stroke volume is equal to the end diastolic volume, the volume of the ventricles at the end of filling, minus the end systolic volume, or the volume left after ejection. So we want this to be around 70 mils per beat. 3 main factors affect stroke volume. The preload, right, what's behind the ventricles pressure-wise, how contractile, how healthy the cardiac muscle is itself, and the afterload, what is the ventricle pumping against. Preload refers to the degree of stretch of cardiac muscle cells before they contract. It's based on something called the Frank-Starling law of the heart. Cardiac muscle cells, just like skeletal muscle cells, exhibit a length-tension relationship. At rest, cardiac muscle cells are shorter than optimal length. Slow heartbeat and exercise increase the venous return to the heart, and this increased venous return distends or stretches the ventricles, just like you would kind of condition an elastic band, and increase the force of contraction. Positive inotropic agents work to increase contractility. So increased calcium influx due to, say, a sympathetic stimulation, or sometimes hormones like thyroxine, glucagon, or epinephrine cause increased cardiac contractility. Negative inotropic agents decrease contractility. So being in a state of acidosis, having too high of a blood pH, too low of a blood pH, having increased extracellular potassium, or taking calcium channel blockers can all decrease the contractility of cardiac muscle. Afterload is the pressure that must be overcome for the ventricles to eject blood. So hypertension, high blood pressure, is the #1 culprit that increases afterload, resulting in increased end systolic volume and reduced stroke volume. If you think about it, if there's too high of a pressure in the aorta or the pulmonary trunk, then when the semilunar valves go to open, it's tougher. And so it takes more pressure of the heart, takes more contraction work of the heart to get that blood out and into those vessels. Sympathetic nervous system activation kicks in under emotional and or physical stress. Norepinephrine or noradrenaline causes the pacemaker, causes the SA node to fire more rapidly, and at the same time it also increases contractility of the cardiac muscle itself. The parasympathetic nervous system opposes those sympathetic effects. So acetylcholine will hyperpolarize the pacemaker cells by opening potassium channels and letting positive charge out. The heart at rest exhibits what we call a vagal tone, which is run by the parasympathetic nervous system. So at rest, we're under parasympathetic cardiac control. So here's just a summary slide showing what different factors can increase venous return, increase contractility, and how that's involved with sympathetic versus parasympathetic. So it's a summary of everything we've gone through. Now, homeostatic imbalances of the heart include tachycardia, or an abnormally fast heart rate, meaning greater than 100 beats per minute, and if persistent, it can lead to fibrillation. Bradycardia, which is having a heart rate slower than 60 beats per minute, and this can result in grossly inadequate blood circulation, but may be desirable when athletes are training for endurance events. Congestive heart failure is a progressive condition where the cardiac output is so low that blood circulation is inadequate to meet tissue needs, and this is caused by coronary atherosclerosis, or a thickening, hardening of the coronary arteries, persistent high blood pressure, or having multiple myocardial infarcts or heart attacks. The development of the heart takes place through embryonic heart chambers, which include the sinus venosus, atrium, ventricle, and bulbous cordus. There's just one atrium and one ventricle to start. And then those develop into what you see here. So that is it for this one. I will see you in the next lecture.